had problems with this at AbSciCons in the past, which I was just telling Nadia about. So in the interest of interdisciplinarity, I tried to go with a simpler, more general title, and I'm going to give, rather than the talk I originally planned, a more of an overview of the research program that we're trying to develop at Georgia Tech. And so my new title will be Stabilizing the Evolutionary Transition to Multicellularity Against Reversion. And so the evolution of multicellularity is one of a very small number of events in the history of life in which the hierarchical complexity of biological systems has increased. And so the others in this very simplified diagram are sort of the origin of life may be considered as one, or the origin of chromosomes, the origin of the prokaryotic cell, eukaryogenesis, then the evolution of multicellularity, and later, in some cases, the evolution of eusociality. And so multicellularity is quite unique in that, unlike many of the other transitions, like eukaryogenesis, which we only know has occurred at least once, multicellularity has evolved independently across multiple lineages. Here in the eukaryotes, it's thought to have occurred at least 25 times. And I think the big question for us in the field of multicellularity research now, that Frank Rosenzweig set up very nicely, is how do you go from a unicellular ancestor of, say, all metazoans to something like a kangaroo? And so I've simplified that process in this 3-part diagram. So our understanding of multicellularity is sort of in its infancy in terms of the process that generates something like a kangaroo from something that may resemble a choanoflagellate. So first, we think it's necessary that you have external drivers that create the benefit to forming a simple social group of cells. So that drives the increased group size from one to many. And then later, there are subsequent social changes associated with the transformation of simple groups to more complex ones. And this could be anything, and this is the part that we really don't quite understand. So I would argue that our understanding of these external drivers and their role is fairly well established. We have good theoretical and empirical results to support things like increased stress tolerance, protection from predation, and improved utilization of public goods as good drivers that might favor the evolution of simple social groups. But what we really don't understand are these social changes that are associated with increases in the complexity of those simple groups. And so, as I may not have mentioned, I work in the field of experimental evolution. And actually, there have been research groups evolving simple multicellular organisms in the laboratory for about 25 years. And this is just a small sampling. Maybe I've captured about half of the instances of this happening in the field. There have been multiple works with algae, both this Chlorella vulgaris in the middle and 2 experiments with different selective pressures with Chlamydomonas. There's also been work done in bacterial systems as well as different yeast model organisms. And so there's just 2 lessons that I want to summarize across these 25 years of history of this emerging discipline. So first, simple multicellularity seems to evolve quite rapidly under the right conditions. So all of the experiments that I summarized here in this slide saw the evolution of multicellular genotypes over the course of a few hundred generations or less. And this suggests that the genetic barriers to making this transition, this first step of the transition at least, are quite minimal. The second lesson from experimental evolution is that simple multicellularity seems to be costly in the absence of these external drivers. So just to pull out 2 examples, in the yeast picture, on top right, there was a 10% cost measured by Ratcliffe et al. In 2012 when their Snowflake yeast system was grown in liquid medium without the external driver, in their case, which was selection for rapid sedimentation. And in the bacteria example, there was a 20% cost measured in Pseudomonas fluorescens, wrinkly spreaders, when there was colonization of the air-water interface was not necessary. And so together, these two lessons point towards a major problem for the subsequent increases in complexity for new multicellular forms, which is that if you have something that's genetically labile and has high fitness costs when the external drivers are not present, you should see the reversion of back to unicellularity should the environment change. And actually, Maria Rebolleira Gomez and Mike Travisano had a pair of great papers come out in the past year where they measured exactly this. So using that Snowflake Yeast system, they first show in growth in liquid without settling selection, that 10% fitness cost that I mentioned. And they also demonstrated that when you do your selections on agar plates, there's an even greater fitness cost for the multicellular forms. So what they found, really interestingly, here we have simple histograms of the area of these multicellular types that form a distribution of clump sizes over the course of 30 days. When they're doing selection on plates, you see rapidly going from the black distribution to the lighter ones. You see size decrease quite rapidly, but when you do your selections in liquid, that size decreases both slower and less pronounced. And so the cost associated with the simple multicellularity actually is a good predictor of how rapidly multicellularity will be lost, as well as the extent to which the phenotype will change. And so we need at least one more arrow in my simple diagram, which is that before you get locked into this positive feedback loop that produces things like kangaroos, you might actually experience an environmental change that favors you exiting this simple system. And so the vicious cycle can't ever occur. You'll never get increases in complexity. And so this is to say that reversion may, in fact, be a major problem that we should consider in the evolution of multicellularity. And so, in my view, there's probably two possible routes to stabilizing the evolution of multicellularity. The first being you can reduce the number of potential reversion mutations. So here I've drawn just a simplest distribution of fitness effects of reversion mutations that I could imagine. So here you have this grayed out dashed unimodal distribution that represents all the possible mutations that will cause a reversion. And the arrow is saying that through evolutionary time, while you're in this positive feedback loop, you may lose some of those potential routes back to unicellularity. And that's just a question about the availability of mutations. Another route would be to reduce the selective advantage to fixing one of those reversion mutations. So if by further genetic interactions and mutations, reversion is no longer beneficial, even when you have environmental change, then that should also prevent the exiting of this cyclical, this cycle here that I had on the previous slide. Or of course, you can do both. And so we sought, our aim was to look for evidence of whether either of these two processes were happening in our laboratory experiments. So we also are working with the Sacrum Icy Service Snowflake Yeast system. And what Will Ratcliffe and Mike Travisano and others did in this original experiment is they subjected a unicellular strain of baker's yeast to selection for rapid settling in liquid media. And over the course of 60 days or fewer, they got things that grew in this beautiful fractal-like pattern. And being from Minnesota, as I already spoiled, they called it Snowflake Yeast, because that was familiar. So the thing that really enables our present study is that the genetics of multicellularity are excessively simple in this system, at least in 5 of the 10 original isolates. The mutation was a single loss of function in a transcription factor called ACE2 that produced the multicellular growth form, and the restoration of a functional copy of ACE2 can produce unicellular progeny. So here's the original mutant, here's a functional ACE2 put into that mutant, And here, if you knock out both copies of the ancestral type, you recapitulate the Snowflake Yeast form. And so we did this from the original 60-day evolution experiment, and just at two intervals. And then we measured a competitive fitness of the unicellular revertants that were derived from multicellular types against their ancestors. And what we saw was really striking is that at the beginning, their fitness is equal to their ancestor. They haven't undergone any mutations. But then by 30 transfers, they were a bit lower, which is the opposite of what you normally expect. And then by 60 days, there was about a 5% fitness cost associated with this reversion mutation. So this is evidence of that second class of mutations potentially fixing. And at least in the case of the the difference between day 30 and day 60, we had a few candidate traits that we thought might explain this decrease in unicellular fitness. So this particular population evolved elevated rates of programmed cell death or apoptosis, as well as increased cell size. And we have these a priori reasons to think that those traits might be costly in a unicellular background, but beneficial for the multicells. So what we really want to know after getting this provocative first piece of data is, what are the dynamics and the consequences of long-term selection on size? And luckily for me, and hopefully for other people, we already had a fantastic postdoc that joined the lab years before me that was conducting a long-term evolution experiment with settling selection, and that is Ozone Buzz, who's here in the audience. And later today, he'll be giving a poster on this long-term experiment and also talking a bit about the effects of high and low oxygen levels on the evolution of multicellular size. So please check out his poster this evening. But what we did is, again, take all of the isolates from this original experiment and perform these genetic reversions on them. And so on the next slide, and I promise, Frank, it'll actually happen on the next slide, we'll these dots that are representing day 200, day 400, and day 600 isolates from two parallel experiments that Ozone had run, will tell you whether or not the transformants with a functional ACE2 are unicellular or multicellular. And so, unsurprisingly, based on the way I set this up, we have some of the populations have actually lost the ability to revert to unicellularity when we put a functional ACE2 back into them. This was particularly validating for us because the two populations, or actually the three populations at the bottom there that were unable to revert also exhibited the most pronounced phenotypic changes over the course of these 600 days of evolution. And one of those really dramatic changes was that the average size of the snowflake yeast clusters increased from 50 microns in diameter to up to 1 millimeter in diameter. And so we did some genome sequencing of these large 600 transfer isolates. Those suggest further genes of interest that might be costly in a unicellular context, but beneficial in a multicellular context. And many of them are associated with the phenotypes that we think are enabling this extremely large size, namely very elongate cells and both polar and side budding. And so if you're interested in some of these new candidate genes and the interaction between those genes, we have another postdoc here, also giving a poster this evening, Tony Bernetti, and his poster will be in the Evergreen Ballroom as well. And so in summary, our results suggest this third model where both the fitness effects of reversion mutations as well as the availability of reversion mutations is changing over evolutionary time, at least for some of our populations. The failure of the functional ACE2 to restore unicellularity is associated with more dramatic phenotypic changes. And we'll continue working on this and be doing some direct pairwise competitions with these more derived strains in the future. So thank you.